This is going to be a live demo. Okay, so I have my, let's see if we can get this working. I have my Dandy Hub up and running. To get the DataBook environment working, you want to go to Environment Management, Dandy, OpenScope.
No, it's already there. So
Oh, it's just one of the kernels that's available? So this, okay. All right, so.
maybe give people a moment if they want to follow along.
Yeah, I'll give you guys a second. So I've Git cloned the DataBook here. Maybe in the future, when you launch it from the website, it might automatically Git clone it, but we'll see if we can get that going.
So do you want people to follow along, or do you want to just show people?
I mean, I guess it's useful to follow along if you guys are trying to get the environment launched. So let me go to this notebook first here. Okay, so picking the environment, which one would it be, Ryan? This one?
No, not that one. Dandy OpenScope.
This one, okay. Dandy OpenScope. All right, I'm going to run this and see if it works. Okay, so this is a notebook we wrote because the Analyst Institute does a ton of quality control, probably more than other people, on our eFIS data coming out of Spike Interface and OpenEFIS. So, I mean, the most important one here is noise labels. So this notebook I made with Josh Siegel because it just goes over the actual quality control metrics that we use before we release our data, and what we would encourage other people to use when they're looking at electrophysiology from Dandy. It actually seems to have a relatively significant impact on the actual conclusions and findings. So that's all just to say that this is important. So I would, even though it's maybe not as sexy as some other models or anything like that. Okay, so one of the most important ones is noise labels. This is essentially, I think it mostly comes from an evaluation of the shape, like the waveform shape for the unit. Well, let's see, did we get this running? Okay. All right, well, while we're waiting for that to run, I'll show you guys on the actual DataBook here. Okay, sorry, I guess I missed a couple important pieces of information. So this data set is the OpenScope's visual coding data set. At this point, the NWB file is a little bit outdated, so it might look a little bit different from the later Allen Institute data sets, like from OpenScope, for instance. But still a lot of really great data in there, and it's used commonly. And it's good for this such demonstration. So the first thing, as a show of hands, how many of you guys have actually so far downloaded NWB files and been able to access the data in them? Okay, great. I don't need to go over this then. So you access the units here, and it has all this metadata associated with each unit. I already went over the noise labels. You can use this basically to calculate the probe yield. And this, I think, varies a lot between sessions, particularly in sessions or NW files that don't belong to the Allen Institute. So this is something that's important to look at. We have the firing rate, clear firing rate distribution. If the firing rate is very, very high, you might suspect that it's not a very good unit. And you can see the log firing rate. I'm not exactly sure the exact cause of this bimodal distribution here, but I would encourage you to look at that also. We have the presence ratio. Amplitude cutoff. So these are types of metrics that I think a lot of people don't really look at, but I think if you do look at it, you'll see there are some clear ways to filter down your units to really, really good, high-quality pieces of information. A couple more, I'll just go over these quickly. Spike amplitudes, ISI violations. This is described here. That's a interspike interval violation. So the way Kilosort works is it identifies repeating spike behavior in the actual direct recorded activity, and a lot of units can have they can have spikes essentially that overlap with one another with themselves. So if a unit exhibits that in Kilosort, you might expect that it's not really a high quality unit or it's not a real unit. Signal to noise ratio is pretty self-explanatory. These other ones, you can look at them on your own if you prefer. But point about this notebook is, these are, like I said, these are important metrics, but not many people look at them. And the Allen Institute has shown, or we've seen, that these have pretty big implications for your findings. So I would encourage you to look at them on your own and actually implement some of this if you're attempting to get high quality findings from these files on Dandy. Yeah.
Do you know any examples of like data sets where this creation does like make a difference in the output? Like, are there any notebooks about that, or does anything come to mind where either doing this quality control or not does make a big difference?
You mean in like the resultant plots, or? Well, I mean, so the Allen Institute, we already kind of do this kind of implicitly. I've seen, like specifically when looking at receptive fields, this is actually really important. Yeah, specifically the noise metric, but a number of other these like ISI violations have shown to be pretty useful in getting good receptive fields, or units that have good receptive fields, that is. Okay. So this is just, this is a basic notebook. I already showed you guys the LFP notebook. Let's see, did this run here? Okay. Okay, another one I want to talk about is opto-tagging. So for those of you who aren't familiar, opto-tagging, so the Allen Institute has transgenic mice, and opto-tagging is a way to identify particular types of interneurons. In this data set, this is an SST mouse, so the cells that you can identify with opto-tagging in this particular mouse would be inhibitory cells. And the way it works is essentially these cells express a particular somatostatin, and this is response. It evokes spiking in response to blue laser stimulation. So you essentially shine a blue laser on the brain, observe activity over time, and you can identify particular cell types. That's the short and dirty version. So this is also, I think this is a visual behavior data set, is what this one is. And the reason I'm showing this notebook is because opto-tagging is important for this type of research if you're trying to identify particular cell types. But the other reason is because it shows, it has a good code example of a type of event-aligned, basically getting event-aligned responses that's used in a ton of different types of analyses, not just opto-tagging. So the first thing here is we have a stimulus table that just contains the information for the optogenetic stimulation, and it includes the laser condition, the duration, the type of laser. And basically in this particular data set, it's basically square pulses of a couple different durations, I think, and cosine waves. You also get the units, which I showed in the previous notebook. You essentially get a huge table of all the different units and metadata associated with each unit, which as outputted by Kilosort. Here is a good example, I think, where we select units based on, let's see. Okay, so I have a couple conditions here which aren't being used, but you could select them based on firing rate, ISI violations, noise, and it might help you pare down your units to particularly good ones. In this case, I'm comparing this wild type mouse with this SST mouse. Okay, and this is the part that is, I think, widely usable across many analyses. You essentially have to pick your resolution to interpolate to, and then you pick your time window from, however long before the event you're interested in, however long after. And this essentially produces this three-dimensional spike matrix where the dimensions would be unit, time, and trial. And then you can aggregate that matrix into getting actual trial line spike counts for every unit. So in this case, we've selected 2,600 units or so across 34 time bins, and you can plot them. So this view I've found to be super useful for many electrophysiology questions. And you can notice here, so these white lines represent the start and stop of the laser. And actually, it's not perfect because we included a little sensor period. The reason for that is because, like in here, these are artifactual. These are not real responses. And these are a result of the laser stimulation. So you actually want to narrow your, the band of time you're interested in a little bit to eliminate those. But you can see pretty clear responses here in the SST mouse and pretty much nothing here in the wild type mouse. So that's a good indication that the data are actually accurate. You can sort them. And so these units are most likely to be inhibitory, and you could sort of find your cutoff visually, but you can also do something more robust. Let's see. So these ones were the ones that we selected to be responsive. The way we did that, I think, was with z-scoring. Let's see, where is that? Okay, no. In this case, it's based on the mean of the response if it's greater than 2, which is basically just a product of the firing rate. So this one was done a little bit, not in a super robust way, but you could add something in here where you're selecting units based on, some deeper property of their firing in response to the laser. And then finally, something about optotagging is that there's not widely used conventions for determining this, right? That cutoff I just mentioned, there's not any really standard convention. So this notebook describes a number of different metrics you could get for each unit, and they're described in greater detail in the citations. But things, relatively simple ones like fraction of time responsive, fraction of trials responsive, but also like KS tests against the baseline and evoked activity. And so this just gives you a little bit of code to get a table for each unit of these metrics. So if you want to do any event-aligned activity for electrophysiology, I would look at this code and you could probably copy it and use it for your own purposes. Oops. Okay. One more notebook I want to show you now is this one. So this was our latest contribution to the DataBook. And I'm showing you this because, like I mentioned yesterday, it's really important to us that the DataBook is growing and has contributions from external contributors. And so this is a really good example. This is probably the best example of that. Guillaume was mentoring Camilla Mora, and she basically produced this really, really good notebook, better than mine, to educate users on GLMs. We have another GLM notebook here, but this one is definitely better and more thorough. And notably, it uses Pineapple, which you guys are now experts in, hopefully, and Nemos. So Nemos, in this case, is the package used to actually train and generate the GLM. So I'll just go over it kind of quickly because I won't be able to explain it as well as the Markdown, but I'll give you guys a summary. So a GLM is essentially a way to model for a given unit, the relationship between the input, the predicting activity, which in this case is white and black flashes on a screen. So it's a way to model the computation that a single neuron is doing in response to the independent variable and exactly how it's producing its spiking behavior. So the assumptions that it makes, first of all, is that there's non-linearity. So let me back up. So the assumption that it makes is that a given neuron is essentially making a computation using some nonlinear function, and like essentially you might look like a waveform of some kind. And then it's nonlinear, and then it also has some randomness, right? So some Poisson activity. So it's actually not that complicated, the basic sort of assumptions that it's making. But I guess it can be a little bit difficult or computationally intensive to train. So Nemos, for instance, is doing a ton of optimization to make it really easy to do this in a large scale way. We have the library set up here. This is also using the visual coding data set. We have all kinds of stimulus here in this file. This is essentially just doing what you guys have seen with Pineapple, reading in the file, getting particular pieces of information, like the unit location. I want to linger on this really quick because in the Allen Institute files, especially the newest ones, getting the location of a unit is a little bit non-trivial. I described that, I think, in some other notebooks. You might be able to search for it, but it varies between file, and you have to cross-reference between the units table and the electrodes table. The electrodes table includes basically spatial and location information for every electrode along the probe, and then the units table only includes indirectly references to the electrodes table. So I could explain that more in detail later if this is a problem you guys run into. But also you could find the code here. So this is getting the location for each unit. You can see here the trial structure for this experiment. A good thing about this one is that it includes many different kinds of stimulus, so it's kind of rich in that regard. Okay, but I think primarily it's interested in black and white flashes in this case, which is the same that's covered in this GLM notebook as well. So you can see you get the input stimulus. This is doing something similar. What we saw, it's filtering by brain area, and it's also filtering by the unit quality. Yeah, these are just zooming in specifically, giving you guys a better understanding of what the inputs are here. But you can see, okay, so now we have rasters and PSTHs for all these different units that are inputted. You can see they look very, very different. You have to choose a bin size, of course. And okay, so this is doing some stuff that's not covered in the, let me actually go down here. Okay. I think a lot of you guys are experienced with machine learning models and these types of things, so you probably already well aware of how to split into train and testing data sets. This one goes over that a little bit more. I think it does something pretty similar or simple with how to split it in this case, but it is an important problem when you're looking at it. So, there's plenty of resources online to do that as well. Okay, to prepare the data for Nemos. Let me see, where am I getting to? Okay, so the main kind of input that it takes, the notebook, this is going to do some other things to the input as well before actually training the model. But essentially, it's getting a window of time prior to the stimulus event that includes whatever your metric of the stimulus is. So in this case, like this GIF here, black, it would be the color of the screen, right? The luminance. And you can get a Hankel matrix, so you would get every binned window of stimulus prior to the spike you're looking at, right? And then this shows you can stack them, right, in a matrix. So this just is a matter of efficiency when you're training. Basically, you're doing a regression. Explain in more detail here. You can see here that this particular unit, most units they show that they behave pretty differently between white and black flashes. It's interesting that a lot of them, you'll see later, they sort of show big activity upon the offset of the flash, which is interesting. Okay, so this is doing another step in order to prepare the input. So this is highly dependent on what your stimulus is and what properties of the stimulus you're interested. or what you think is most significant, or what you're hypothesizing is most significant for the activity of the population of cells you're looking at. In this case, it uses this raised cosine linear basis. I'm not familiar with all the logic and how they decided this particular linear basis, but there's also, there's discussion here exactly why you'd want to use this and how. In a really simple GLM, you don't need this at all. It depends, right? So in the example shown above, here, you might not actually need it depending on what cells you're looking at, right? It's pretty clear, but in this case, this is the one they chose. Okay, yeah, so this is basically convolving this linear basis over the input variable that they're interested in here. And finally, you can actually run it with Nemos really simply. So everything else above was basically sanitizing your inputs, preparing it for the form that it's required for GLM, and then you feed it in here, right? It's really simple. And you can fit it. Let's see where it's shown here. I guess there's no output filter shown, but what it's doing is it's training this output filter I mentioned earlier, which can be convolved across your input stimulus, and ideally, it will predict the spiking behavior of the unit. So let's see how well this one performs. You can get the actual PSDHs and the predicted PSDHs, and this one works very well, it seems like. So I mean, I think you normally would, for predictions, because there's it introduces this Poisson, this randomness, you'd probably, for your prediction, run many different sort of simulated prediction trials here. Not sure where it does that, but essentially, I mean, you can see that this works pretty well, and in the code-wise, it was actually pretty simple, in addition to all the preparing your input. So another thing that Nemos offers is this population GLM, so it works for a huge population. It's optimized, so it's not doing redundant computation, and you can see it works very well for many units in this case. So it's not just for particular units, and it gives you a really good idea of exactly how these units' behavior vary in response to the input variables, which in this case is the black flashes and white flashes. Okay, there's two more parts here. This one I'm not as familiar with, but so it's called coupling, and what it's doing is it's using an additional predictor, which is the behavior of the units nearby. So the nearby connected units are supposedly related to the activity of a given unit, and I don't know the math about how it's doing that, but Nemos provides the ability to do that as well. And it's plotted here, and then I think there's one more spot. Oh yeah, then you want to also evaluate their performance. So you can use R-squared value. That's what we use in this notebook as well. I'm not sure if this goes over other ones. Okay, yeah, the pseudo R-squared McFadden. So this is definitely something that's essential you're going to want to do if you're making a GLM. And when I was writing this notebook, this was absolutely essential, is using robust metrics to evaluate their performance. I mean, in this case, you saw that it had pretty good predictions just visually, but you definitely need to make sure you decide on this early on and make sure that it's robust and use it thoroughly. There's a couple other notes here. I would just encourage you to look at this notebook. It's just a really, really good example of how the DataBook can improve the ecosystem, because when I was writing the other GLM notebook, the documentation on GLMs, despite the fact that it's a really, really powerful analysis, was pretty poor, and a lot of the technical details were kind of glossed over in a lot of the online documentation. And so this notebook is, I think, an excellent addition to the ecosystem, and I think there's a lot of other fruit in the ecosystem that could be harvested in this regard. Like, there's so many other models that aren't really covered on, like it's one thing to learn about how to do a good model from your PI, right? But if you had a platform like this where anybody can learn this model really, really well with a ton of detail and code and, actually replicating the data, it's super, super valuable. So I wanted to show this notebook as a great example, and thanks to Guillaume and Camilla for adding it. Okay, I don't have any, there's other electrophysiology notebooks here, but you guys can look at them on your own if you're interested. I don't have anything else for this particular case. And I believe that this environment should be working now. So let's try it out together. Okay, any questions? All right, thanks.